So now, so you have these three stop codons. In the termination process, you have these three stop codons. So as soon as you reach these stop codons, they are not recognized by any tRNA. And then you have some release factors that come and bind to this particular stop codon, and then your whole peptide is released um, from the P site. So you have free polypeptide, and then the whole assembly breaks and is ready for another cycle of or to go and bind to another mRNA. I should have mentioned that in this elongation process, also there are a lot of factors involved, which are called elongation factors, and they are also important for this movement of this initiator complex, because anything that moves in biology, it needs energy, right? So they are a class of GTPase enzymes, which help this whole assembly to move one codon at a time, right? So they are also important in this translation process. Okay, so now we'll talk about Flexizymes. Okay, so these are modified amino acid synthetase. So this is one of the very important amino acid synthesis that people discovered or maybe made using a library of RNA, which was called R24. And this can particularly load different amino acids, like which are not really proteogenic. So these are called non-proteogenic amino acids, right? So or maybe just non-proteogenic substrates, right? Not even amino acids. So they have different groups present on them, like which are not generally present on different amino acids. So this looks like a phenylalanine, but there is this particular group which is not in phenylalanine. Okay, so they also modified this particular R24 enzyme, made it smaller, right? So they can incorporate this different non-proteogenic substrates. Okay, and the reason that we want to do that, why we want to incorporate something else other than methionine, is because it really diversifies the library that you are synthesizing. You can incorporate different like different non-proteogenic substrates. Also, you can incorporate some fluorophores, which are very important in recognizing a particular protein or a peptide if you're doing some assay with it in the future, right? So these are very important. Now,
Hello.
this is an example. Yeah. Yes,
I have a
Anish.
doubt from the
Yeah.
previous slide.
Okay.
So here we can see the tRNA ASNE2,
Yes.
and there is this like on the right hand side, it's tRNA ASNE2. So
Yes.
like the very first picture, it shows like molecule which has like your R24 library.
Yes.
So is it like you have screened it from like a pool of library which
Yes.
where okay, so
Yes.
what is on the right hand side, like what are these modifications?
These
If
modifications, so they just recognize that you don't need this long, like, so there is this dot dot dot, right? So
hmm,
these are
the
also
variable sequence.
variable sequences, but
Hmm.
they understood that they don't need these extra sequences.
Okay.
Yeah, so they are not really helping in any way. It was just a random library, so they just narrowed it down to these different groups which are smaller, right? So you don't want to synthesize which is very big and it's gives you the same efficiency as a smaller thing. So
Yeah, but then
yeah,
my question is like,
yeah,
why are you even modifying this tRNA? Like, I
yeah,
understood it's
yeah.
like you're reprogramming the like tRNA,
Genetic,
but what is
yeah.
the use of this?
Yes, so it's a very good question. So the use for this is, so you have those 64 codons, right? Different codons which code for different amino acids. And now you have only 20 amino acids, right?
Mm-hmm.
So there are some codons which repetitively code for the same thing. Do you get my point? So you have phenylalanine, which is coded by 3 different codons. And they code
Yeah.
for the same phenylalanine, right? But what if you can just two redundant codons which code for the same thing, and you can incorporate something else. So that way you can diversify your library.
Okay, so another question, why do you
Yeah,
want to, like, generally you choose a molecule from the library for a
yeah,
specific like specific purpose,
yes,
but here why are you like further diversifying it?
yes.
Like, what is the
So
objective?
yeah, so the objective is when you are going into drug discovery, you want to test as many molecules as possible.
Okay.
So
Mm.
there are like, so when you are talking about protein-protein interaction, or some molecule going and stopping some interaction, so you need molecules that have a very high binding affinity, right? So for agonist or antagonist type of molecules, you need a very high binding efficiency. So
Okay.
for increasing the binding efficiency, you want to test as many molecules as possible.
Cool.
Yeah.
Mm.
Okay. So yeah, I mean, I'll show you an example that how you can actually make molecules and test them. Okay. So that's why they are using this Flexizymes to make different libraries and diversify the molecules for drug discovery process. Okay, so now, so this is a typical codon library looks like. So these are the stop codons that they have not shown what they code for, right? Because they are stop codons, they don't code for anything. They just stop the translation. These other codons, like you see this leucine, it really has like 6 different codons which code for the same thing you see, right? So these are redundant. They are repetitive. So these particular different codons can be used. So when you synthesize using this particular codon library, you get this kind of molecule, right? Which is a simple system, right? But if you modify these particular codons, like they have modified this phi codon to methyl phenylalanine, okay? So you have N-methylated phenylalanine now in the system. Similarly, they have changed some of these codons like methionine serine, right? So this methyl serine. So this N-methylated serine, then you have N-methylated glycine, and then the most important one that we talked about. So we talked about introducing different non-proteogenic residues at the place of methionine, right? So here you have this chloroacyl D-tryptophan. Now, incorporating a D amino acid also changes the stereochemistry of the whole library. So this is again expanding the um, the traditional molecular libraries and then just taking it to another level, making a lot more a lot more diverse library than what you used to have before. So they made this particular peptide. Now, this is a chloroacyl group. It's highly reactive. And you have a cysteine at this penultimate end of the peptide. So when you have a cysteine residue, you have a sulfur attached on the side chain. So that attacks over here and you get a cyclic peptide. Okay. So generally, when we are talking about drug discovery, peptides are highly, okay. Uh, okay. There was some message. Um, so now this particular peptide library, you can cyclize it, right? Now the reason why we want to cyclize is because most of the proteins or peptides, they are highly exposed to proteolysis. Okay, so you don't want your drug to be proteolyzed and broken down into smaller amino acids, right? Because you want to keep the conformation and also the structure of the protein so that it can act as a drug. So that's why you want to cyclize those peptides so that you can reduce the accessibility of these proteases to the peptide. So this is just a collection of all the different like non-proteogenic substrates that you can add at the place of methionine. And so it's quite a lot. You can add different residues, even you can add different peptides like tripeptides or dipeptides into the N terminus of the protein. Okay.
Hello.
And yeah,
Uh, sorry. So
yeah,
does
no
these
problem.
you're putting all these modifications in there. So
Yeah.
does this words flexible has to do something with the conformation of your tRNA? Like, does it make it more flexible or like?
Yes. So the Flexizymes that we talked about, they are generally incorporating all these different non-proteogenic substrates onto the tRNA. So those are more important than the tRNAs, because tRNAs is the structure is the same. We're just modifying the amino acyl RNA synthetase, that because we want to incorporate all these different non-proteogenic substrates. Does that answer your question?
Partially,
Okay.
but I wanted to know whether did you. So you showed that tRNA
Yes.
sequence, right? But
So
like, if I missed,
this one.
did you show any Flexizyme?
So
Yeah.
this is the particular Flexizyme. So
Oh, acha.
this is the
Okay.
tRNA and this is the
Okay.
Flexizyme, right? So this particular
So
tRNA, which
all
is
these
unmodified.
modification is being done by the Flexizyme on the tRNA, or
Yes,
okay, all
yes,
right, okay,
right.
yeah.
So these Flexizymes, yeah, I was not very
Mhm.
clear about that. Yeah,
Okay,
thank you.
thank
Yeah,
you.
yeah. Okay, so you get this. So we are talking about this. Yeah. So yeah, so you have all these
Hello.
different non-pro. Yeah.
One thing that you told that in the previous slide, that you, if there's, you add a methionine there, you, if you add a methionine in the fast of any, oh sorry, methyl group any fast of any nucleic acid, then is do it more diverse. But here it's, how did we more diverse? Because these are just replacing one amino acid to another.
Yes, right. So what is happening here is that you have this normal library, right? Now, to diversify this library, so these are all redundant amino acids. Like they are repeating the sequence, and they are producing the same amino acid, right? For different sequences. But we want to make it diverse by introducing this. So this N-methylated serine has a different function, right? Than just the normal serine. So it makes the peptide more hydrophobic, which is good for drug development. So most of the drug molecules that you see have a very high hydrophobicity. Okay, so
Okay.
this particular methylserine provides additional hydrophobicity for the peptide. So now you see that when you have a peptide bond, you have a NHC double bond O, right?
Yes.
So that hydrogen attached to the nitrogen, that forms hydrogen bonds with water molecules around it, right? And when
Yes.
it does that, it becomes more soluble, because more hydrogen bonding with the water molecules, more interaction, so it becomes more soluble. But you want your drugs to be a little bit more hydrophobic, so you introduce a methyl. Now it doesn't has any the it has a one less um, hydrogen bond, right? So that decreases the solubility,
Okay.
and also the hydrophobicity
Okay.
increases, right? So you want to do that. So this is how you incorporate different amino acids which are non-proteogenic. These are the different classes. Now this one is really important that we discussed just right now, that you have a chloroacyl group on the end term of this amino acid, like this tyrosine, and that helps in cyclizing. So this is tryptophan, chloroacyl tryptophan, and methylated one, right? So this helps in cyclization of the peptide. Okay, so this, okay, just a second. So this particular approach is called flexible in vitro translation. So because they are using Flexizymes to incorporate these different non-proteogenic groups, and then so this is called flexible in vitro translation. Now, how do you use this for drug discovery, right? So when this in vitro translation, flexible in vitro translation, is coupled with mRNA display. Okay, so I'll discuss in this particular schematic diagram what mRNA displays. But so here you have incorporated this particular FIT and cyclization in the process. Okay, but if you just want to have a look at mRNA display, you just get rid of this translation, just FIT, like flexible in vitro translation and cyclization process, and you get a normal mRNA display. But coupling this, they have used this to find some non-standard peptides that are helpful in stopping some protein-protein interactions with very high affinity. Okay, so you have this DNA library where it undergoes transcription first. Okay, and then you add a puromycin ligation, ligated tag, right? So you have a puromycin molecule at the end of this particular mRNA. Now this undergoes translation using the flexible in vitro translation system. And then because you have that chloroacyl group, it cyclizes itself into a macrocyclic peptide. And then you have reverse transcription. So this mRNA again goes back, and this, it is still, you can see in this schematic diagram that it is still attached to the particular peptide, right? So you know that which mRNA belongs to which peptide. This is a very important thing to know, that which mRNA displays which peptide. Okay. So now this particular library undergoes reverse transcription. This mRNA is again back to its cDNA, complementary DNA fragment, right? Now you do a affinity test. Okay, so you just expose all of this library to immobilized proteins or immobilized target molecules. Okay, and they go and bind to this particular molecules. Then you wash off the peptides which did not bind, so you don't need them. Then you use this. So you use recovery. You use the recovery process using PCR, where you cleave this puromycin tag and use the cDNA library again to do the same thing, right? So you now use a particular cDNA library which is being selected, which binds to this particular protein, right? So this peptide particularly binds to this protein. You know that it binds with high affinity. So you again expose this to the cDNA library. You recover this particular DNA library, and then again do the same process for 5 to 6 more times, and then you get a particular peptide which binds with very high affinity with this protein molecules. Was this clear? Was I clear about this? Any
I have
questions?
a question.
Yeah, sure.
So, is it like first you have those peptide libraries you made earlier, like
Yes.
in relation with those tRNAs?
Yes.
Then you go on to like do a screening for mRNA. So, what is that starting point? Like, how do you?
Yeah, so you start from here. So you have this random DNA library.
Okay, yeah, these are the random sequences from which you further enrich it to be specific for your modified tRNA.
Yes, right. Yeah, right.
So it's basically you have formed a library, then you're enriching another library. Is it like correct?
So out of this library, so there are a lot of random sequences in here. So you have a lot of N's. N means it can have anything like A, T, G, C, like anything, any of those 4 codons. Now, when you just make a mRNA library out of this, you get different codons at different positions, right? And once you translate them, you have different amino acids at different positions. And this particular mRNA library is attached to those peptides, right? So you know that where it is coming from. Okay. And now once you have this peptide, you just expose the peptide to this protein molecule, and it's still attached. The DNA library is still attached to this peptide, right? So only those DNA libraries will remain for which the peptide binding affinity is very high. And in the enrichment process, you are just amplifying more of these particular peptides.
Yeah, sort of you're screening the library for the ones which are specific for your tRNA.
Yes, right.
Yeah. Okay.
So using this, actually, so you can't even imagine what size of library they can screen using this, because
Exactly.
it's around, yeah, it's around 10 to the power 12, which is a very high number of molecules,
Exactly. Yeah.
right? So you can test that many molecules using 6 different cycles, and it's a very quick process. So
Prabhat,
you, yeah,
yeah,
Sunil, yes,
Prabhat, I have a question relating to affinity selection.
yeah, sure.
So, what are these substrates particularly? Like, is it chromatography technique like which you are using to immobilize the thing, or
So that's a good question. So we immobilize these proteins using streptavidin and biotin tags. Okay, so on this particular protein, you will have a biotin, and on the immobilized membrane, you'll have streptavidin. So
is a
that biotin, yeah. So biotin and streptavidin is known to have highest affinity of all biological molecules.
okay?
So they have a affinity of around some picomolar range. Okay, so that's very hard to find. So they generally get immobilized using that. And then when you expose the peptide to this particular protein, they are also immobilized on the surface, right? So they won't move because they have some affinity to this protein. And you can wash off the other peptides that don't bind, right? So you just keep it for some time, and then you wash it away. So it's very similar to what you do in
Northern
affinity
blot,
chromatography. Yeah, I mean, you can do the similar thing in affinity chromatography, I would say.
okay,
So in
yeah,
affinity chromatography, you have those histidine tags, right? And then
yeah,
they go and bind to the particular substrate in the column, right? And then you can cleave it off using butazole later on, right?
mm,
So it's similar to that, I would say. Yeah. Okay.
yeah, it's clear.
Yeah, it's clear, right? So now you can get some inhibitors or agonist or antagonist using this particular approach, which bind with subnanomolar capacity, and that's very important, right? So you want drugs which bind with very high affinity, so that when you administer the drug, you have to give a very small amount to cure the disease or to stop any interaction, right? Okay, so now you have all these different approaches which are similar to this mRNA display. So this is just mRNA, plain mRNA display, but the technique we discussed before was a rapid technique, where you fuse mRNA and FIT together to get the different libraries. But you have this phage display library, which was awarded Nobel Prize in 2018. You also have this one bead, one compound library, which our lab works upon. So this is something that we work on. We use this process to find different peptide molecules which bind with uh, target protein with very high affinity. Then you have this east display technique, which is a very recent one. It's been like I think it was this developed in 2014 or so. So these are very recent technologies that you have for screening different peptides. So just wanted you to know that you can read upon this if you're interested. You can just go through. And with that, I think that's all. Thank you for listening, and thank you for inviting me uh, for this ISERC IO. It's been a pleasure.